Hello everyone, my name is Mateus and this project was developed at LMMP PUC-Rio with Georgia Avendano and Professor Márcio Carvalho. The name of this project is Controlled Release of the Inner Content of the Gellan gum microcapsules using temperature as the trigger mechanism. The summary for this presentation, we have an introduction and motivation, the main goals of our study, the composition and production of our samples, the experimental setup where we heated our microcapsules, the trigger events observed, the results and discussion, and the final remarks at the end. While microcapsules are formed by a core material involved by a membrane, also called as a shell. Its size varies between a few millimeters and they can have a single core, as shown in this image, or multiple cores. They have an important role in food and pharmaceutical industries due to their capacity to improve ingredients, handling, to mask undesirable flavors, odors, to protect different components from chemical or physical degradation during storage or after ingestion. Thus, the technique of microencapsulation depends on the physical and chemical properties of the materials to be encapsulated. The use can be extended to different applications that require the protection of the interface and its controlled release. Different triggers can be applied in order to activate the core release, depending on the application and capsule's materials such as temperature, pH, stress, osmotic pressure, UV light, or chemical reaction. Many applications of microcapsules require them to stably encapsulate their contents, releasing them only after exposure to a specific stimulus. An important class of stimuli is the change in temperature. This finds application in transporting and releasing food additives, cosmetic, or drugs, which often require release to be triggered at body temperature. This example below is showing the effect of thermal trigger on a microcapsule. When it is under a heating process, the membrane is broken. Consequently, your content is released. In addition, recent studies have shown advances in the applications of microcapsules in the process of enhanced oil recovery as fluid mobility agents that can lead the flow to a total redistribution. Also, they can carry chemicals that could react and clear the flow path. In this work, we present a study with a biopolymer substance that does not harm the environment involved. The main goal of this project is to study the microcapsule's membrane behavior when exposed to a heating trigger process. The methodology in this work analyzes the influence that the release event have with the microcapsule's properties such as diameter and thickness of the shell. Also establish time and temperature conditions at which the inner content is properly released. In this work, we approach two different methods. The first one consists in varying the temperature between room and trigger temperature. The second one, we work with a constant temperature. Basically, the microcapsules studied in this project are based on the composition of double emulsion oil in water in oil that undergoes a gelation process in its production. Its structure is divided into three phases: its core, that is composed by sunflower oil, as well as its continuous phase of production, but with an addition of calcium acetate. And finally, Gellan gum makes up the shell phase structure. This biocompatible gum acts as a stabilizer, thickening, and galling agent in a wide variety of products, and is capable of forming hydrogels when in contact with cations. Also, this biopolymer is a thermal-responsive agent that does not harm the environment involved. After the production, the continuous phase is changed from sunflower oil to acetate buffer in a recovery with stabilizer process. The capsules are produced with microfluidics, in which each device allows the production of oil and water in oil templates with turnable mechanical properties, and the dripping regime, as you can show here, is desired in order to achieve the best condition of production. This is the experimental setup, where we have an inverted microscope to observe the capsules in real time. We also have the heater, which has the function of heating the heating fluid, and we use the deionized water to circulate in our experiment. In addition, we have a thermocouple that will provide us with temperature data over time. We also use the computer that processes all of these data. And the microcapsules are visualized through the support plate, which we can have a variety of samples tests at the same time. Now let's talk about the temperature trigger events. On the slide, you can see the change in temperature behavior of the capsules. In this first video, we see the structural alteration when the heating trigger is activated, as well as the movement of the internal oil when encounters with the acidic medium of our continuous phase acetate buffer. In the second video, we have a more detailed view of the membrane breaking. One promising explanation to this phenomenon is the polymer hydrolysis, in which at some trigger temperature under acidic conditions produces monosaccharides by breaking the glycosidic links between the monomer units in the structure of the molecule. And in the latter image here, we see the coalescence of the oil after its release. We quantify the interface release and study the effect of constant temperature by counting the number of remaining capsules as a function of time and temperature set. As we can see here in the graphic, between 70 and 74 degrees Celsius, a small number of capsules are destroyed. And on the other hand, 75 degrees Celsius proved to be more viable for the content's total release, where we can clearly see an abrupt drop in the number of remaining capsules, acting as an instant stimulus to this temperature level, leading to a total degradation. The tests below 70 degrees Celsius showed up to be inconsistent and with a random behavior with the stimuli. So after this test, we establish an optimum trigger temperature, that this is the 75 degrees Celsius for the Gellan-based shell, that can guarantee us the inner content release. As we were able to guarantee that 75 degrees Celsius would deliver our encapsulate component, we then studied how time at this optimum temperature is affected by the properties of the capsules. The idea here is to analyze different diameter groups of the microcapsules, and for each one of them, we aim the same thickness diameter ratio for the samples. After all data analysis, we registered few differences between the two groups. For the first one, the particles with the smallest TD radius, here in blue, had rougher response to this temperature trigger mechanism and needed almost 6 minutes for the full release of its content. But this response did not mean an early trigger, because if you compare with the bigger radio ones, orange and yellow here, the first responses for this trigger outcome from them regarding their subtle and release throughout time. Therefore, from this test, we could not relate an early trigger with an easy content release. Yet we did observe effectively more time to disintegrate these bigger radio ones. In the second group, down below, shows a similar behavior. We can observe an abrupt drop in the percentage of the remaining capsules for the smallest TD radios, here in blue, as with group 1, but an increasing full release time. As for bigger radio ones, in orange and yellow, the release is rougher if you compare it with the first group, yet more subtle than the smallest in this group. Now changing the temperature control method. In this test, we work with temperature variation starting at room temperature and ending at the optimum one that we find down in the past slide. The idea here is to compare the capsules with the same percentage of thickness but with different diameter sizes, and see if there is a difference between the behavior between them. Here we can see that both reached 75 degrees Celsius at the same time, but the larger one, here in purple, we have an instantaneous response and lost its membrane, while the smaller one, in blue here, took longer to suffer from the heating process. And finally, we did some aging comparison tests with single capsules. The objective here was to evaluate how the aging time of the microcapsules affect the cargo release. They were performed using capsules produced more than a year ago and freshly produced capsules of the same properties. Experiments were performed varying the external environment temperature from room temperature, 25 degrees Celsius, to 75 degrees Celsius. When the temperature reached 75 degrees Celsius 10 minutes after the beginning of the test, then we can observe the first major changes in the membrane structure and ending with a film formation. There was no big difference between the old and the newer ones. And finally, the final remarks here, the results of this investigation show that the inner content release can be a function of the environment temperature and the microcapsule's properties. We have reached an optimum trigger temperature where we could successfully disintegrate our biopolymer shell. And in this work, we analyzed the low acyl Gellan form capsules, and a promising study seems to be a comparison with the natural, elastic, and soft form of the high acyl Gellan form. And another promising lead is to raise the percentage of the thickness of the shell. And thank you all for the funding agents that support this work, and thank you all for your attention. Thank you, everyone.